Good morning, everyone. Welcome to our second virtual field trip in our soil and water conservation series. My name is Chandler Rell. I'm the soil and water conservation instructor here at the University of Arkansas. I want to thank you all for joining us today as we dive into the science behind soil and water conservation issues in the state of Arkansas, particularly row crop production. Before we get started today, I also want to thank the USDA and NRCS for their funding and their collaboration in this project. We're joined today by Drs. Kristofor Brye and Dr. Della Lunga. Dr. Brye is a university professor in applied soil physics and pedology, and Diego is a postdoc serving in Dr. Brye's research lab. So good morning to both of you.
Good morning. Thank you, Chandler, for this opportunity. Diego and I are greatly appreciative for this opportunity to contribute to this virtual field trip series and give a little bit of insight into some of the research and techniques that we've been using to conduct our in-field research over the last few years to a little bit more than a decade.
Great, yeah.
Pleasure to be here. Thank you, Chandler. Dr. Brye and I are very excited to have the opportunity to show how management practices can address conservation issues in our agricultural fields.
Absolutely. Well, we're happy to have you both here. For those of you that are joining us live, feel free to drop a question in the Q&A function throughout any point in the presentation, and at the end of the presentation today, we will try to answer as many of those as we can. But with that, I think we can go ahead and get started. First, today, we're going to be hearing from Dr. Brye about soil and water conservation issues in Arkansas. Now, Dr. Brye has been a professor at the University of Arkansas since 2001, and his research has covered many different topics of soil science. But most recently, he has focused on the intersection of soil conservation, field management, soil physical, chemical properties, and how they relate to trace gas emissions in Arkansas row crop production. So in a little bit, we're going to hear more from that from Dr. Brye. But first, I want to ask you, why does this all matter?
Well, thanks, Chandler. Conservation does matter. Conservation is extremely important because there is no replacing productive soil, high quality air, and water resources. We just don't have a substitute for these foundational resources that all of agriculture everywhere is built upon. One of the biggest discrepancies, for example, when it comes to the soil, is how long it takes to make productive soil versus how long it takes to degrade productive soil.
Absolutely.
Degrading productive soil can happen in an instant, literally seconds, from a massive rainstorm, runoff, soil erosion. But to make healthy, productive soil, we're talking hundreds of years at a minimum. So we need to make sure that conservation is at the forefront of all agricultural activities that we do throughout the state and beyond.
Absolutely. Well, that all sounds very interesting and great to me. So let's go ahead and see what Dr. Brye had to say about soil and water conservation issues in the state of Arkansas.
Good morning. Thank you for that introduction. Today we're north of the University of Arkansas campus here in Fayetteville, Arkansas at the Milo J. Schultz Agricultural Research and Extension Center. I want to talk for a little bit about some of the conservation issues that are affecting Arkansas in agriculture these days. And then I want to go into a little bit more detail about what we're doing with one of the most recent conservation issues that is facing Arkansas agriculture presently. So to start with, soil erosion. Soil erosion is a big problem still today in Arkansas. It's been a problem in the United States and many other places for decades, going back to the Dust Bowl years. We need to keep soil in place on the upland landscape, and we need to try to keep it out of our waterways, where that soil tends to end up after being eroded away. We want to do everything we can to keep clean water in our waterways and avoid turbid water. Turbid water is extremely hazardous for aquatic organisms. It covers plant leaves so they can't photosynthesize. It chokes out aquatic organisms that are attempting to gain oxygen from that water. So soil erosion is still a problem that is facing Arkansas agriculture these days. Another important conservation issue that we have relates to a really important commodity that Northwest Arkansas here is got a little claim to fame for, and that's poultry production. Poultry production is concentrated here in Northwest Arkansas, and with 20,000 birds or so in a poultry house, there's a lot of waste materials. That waste material traditionally gets applied to the land surface for its nutrient value. However, we've ended up with the circumstance where there is a tremendous nutrient excess here in Northwest Arkansas. Contrast that with Eastern Arkansas, the Delta region where our row crop agriculture occurs, and for the most part, there are nutrient deficits. Producers have to use lots and lots of fertilizers every year to maximize yields. So we've got this big nutrient imbalance geographically, Northwest Arkansas versus Eastern Arkansas. That's a major conservation issue that we're trying to deal with these days in the state. With both soil erosion and nutrients, if they run off into the nearby water bodies, streams, lakes, ponds, that sort of thing, we've got water quality issues that develop. And so water quality is a conservation issue we've been working on for years as well. We want to keep the sediments out of the waterways. We want to keep the nutrients as much as possible out of the waterways, particularly phosphorus and nitrogen that end up running off with sediments and finding their ways into the nearby streams can go through a process called eutrophication. Eutrophication starts with excess of biomass that's produced from algae. When that algae dies after using all of the nutrients up, now that material starts to decompose and the oxygen in the water column is used up. The end result of eutrophication is often fish kills and devastation to the aquatic ecosystem. So we need to keep the sediment and the nutrients out of the waterways from a water quality issue. Not only do we have water quality issues, we have water quantity issues. Now that's a little counterintuitive for a state that generally has abundant water resources, but it doesn't rain when we want it to all the time. Sometimes it rains when we don't want it to. But to produce high quality yields in our row crop delta region of the state, we need to use irrigation. Much of our irrigation water comes from groundwater and groundwater aquifers in the eastern Arkansas third of the state. The problem from a conservation standpoint is we're pulling water out much faster than that water is able to recharge the aquifer. So we've got this massive imbalance in withdrawal rates compared to recharge rates, and that's causing the water table to drop. Got to drill wells deeper and deeper every year, and that becomes more and more expensive. So we want to make sure that what we're doing agriculturally, scientifically, is trying to conserve water resources so we have a future of irrigated agriculture. The fifth and final conservation issue that I want to spend some time talking about relates to air quality. We've identified soil and water related issues, but also air quality is something that's important these days. And I'm specifically referring to carbon dioxide, methane, and nitrous oxide that's produced by the soil and is emitted into the atmosphere. Now, all three of these gases are produced naturally, all across the landscape. Soil will breathe just like you and I are breathing right now. We take in oxygen and we expel or exhale carbon dioxide in the process referred to as respiration. So soil respiration and the production of CO2 really happens everywhere in the landscape nearly constantly. That's a little bit different story with methane and nitrous oxide. They have a lot more spatially isolated pockets of the landscape and some specific conditions that have to be met before those gases are produced by microbial activity. So for example, methane. Methane requires an extended period of prolonged soil wetness, saturated conditions, maybe even ponded conditions, before carbon dioxide switches from being the main gas that's produced to methane being the gas produced. Now this happens naturally in wetlands and low points in the landscape, maybe in riparian areas along streams and rivers. Nitrous oxide, similarly, has a very spatially isolated place in the landscape that requires certain conditions. The first precursor to nitrous oxide emissions is having a source of nitrate nitrogen that is available to go through the process of denitrification. Now, denitrification is a natural process. It happens periodically in actually many places in the landscape. If denitrification goes completely to the end product is dinitrogen gas, N2. That makes up 79% of our atmosphere, and no harm done. But when denitrification is short-circuited and stops partway, then we get nitrous oxide, or N2O gas, that's produced. The problem with all three of these gases, and that's where the air quality issue comes in, is when these gases end up in the atmosphere, they're actually all really good at trapping outgoing heat radiation that's emanating from the land surface and trying to escape back into the outer reaches of the Earth's atmosphere. So when the heat energy is trapped in our atmosphere, well, that tends to increase air temperatures. We can't do a whole lot about carbon dioxide. We can some, but methane and nitrous oxide, they're particularly damaging in the atmosphere because on a molecule by molecule basis, methane is about 30 times more potent than every molecule of carbon dioxide. Nitrous oxide on a molecule by molecule basis is about 300 times more potent than every carbon dioxide molecule in the atmosphere. So what we need to do is be able to understand what some of our agricultural management practices and decisions are that affect the production of these gases and their release into the atmosphere. So let me go a little bit farther with this. About 15 years ago, the United States Environmental Protection Agency specifically identified flood-irrigated rice as a major and significant contributor to methane emissions to the atmosphere. Well, it turns out that rice production is one of Arkansas's leading row crops. Arkansas is the leading state in the United States for rice production pretty much every year, has been for a long time. So the fact that rice, flood-irrigated rice, was identified, that necessitated me changing my research program to start looking at methane emissions from flood-irrigated rice. And that's exactly what we did. So rice is typically grown by establishing a flood about a month after planting, and that flood stays on for several months, only being released to dry the soil out prior to harvest. So what happens is the soil gets really wet, saturated, and loses its oxygen pretty quickly, about 2 weeks after the imposition of the flood. That changes the environment from carbon dioxide being produced to methane being produced in this flood irrigated production system. So what we wanted to do was go in and identify some places, management practices where producers have decisions. You could do one thing or you could do another. We could plant rice here or we could plant rice there. So let me summarize some of our research results in a qualitative manner. We first started with soil texture. We've been able to demonstrate through field studies that rice produced under flood irrigated conditions on a silt loam soil would produce greater methane emissions than on a clay soil. We moved to nutrient sources. Nutrients can be applied in organic form or inorganic form. We've demonstrated from field studies that when a large carbon-containing organic nutrient source, like poultry litter in fact, is applied to a flood-irrigated rice scenario, you can expect greater methane emissions than when inorganic fertilizers are used. We moved then to water management. Now there are a couple variations of flood irrigation. Continuous flood is the most popular traditionally and still these days, but there are a few other alternatives that actually drop the flood periodically for a variety of reasons. There's an alternate wetting and drying scenario where the water is allowed to evaporate, and then when it dissipates, the flood is put back on. There's intermittent flooding, and then there's another system called mid-season drain, where partway through the rice growing season, the flood is completely released, the soil's allowed to reoxygenate, and then the flood's put back on. So we've been able to demonstrate from field studies that continuous flood irrigated rice, we can expect greater methane emissions compared to these other alternatives, even to the flood irrigated water management scheme. One of the last production practices that we've studied is rotation. Rice is grown in typically 2 rotations. There are others, but mainly continuous rice, where rice is grown every year, or rice is grown in rotation with soybeans. So when there is a large residue producing crop like rice the year before we produce a subsequent year's rice crop, we can expect greater methane emissions than when a low residue producing crop like soybeans are produced the year before. So through field studies, and we've done some greenhouse work as well, we've demonstrated a number of different management practice options that we know exacerbate or increase methane emissions compared to those that might reduce or minimize methane emissions. So with the potency of nitrous oxide, we obviously had to add that into our suite of measurements too. So we started simultaneously measuring methane and nitrous oxide along with carbon dioxide production and release from the soil environment. And we've done some greenhouse work where we've identified that the application of nutrients, the timing, if there is 1 or fewer split applications of nitrogen, that would produce greater nitrous oxide emissions compared to 2 or more split applications of nutrients. We give it a little bit at a time, the plant can use it, we don't have nitrate nitrogen available to be denitrified. Well, we also studied in the greenhouse soil moisture conditions, and we've identified that if you keep the soil as uniform and stable of a water content as you can, we're going to minimize nitrous oxide production instead of having the soil moisture bounce up and down from wet and dry, wet and dry, wet and dry conditions. We've studied in the field tillage effects on nitrous oxide production and have demonstrated that conventional tillage disturbing the soil in the spring prior to planting rice would tend to increase nitrous oxide emissions to the atmosphere compared to a no tillage setting when the soil's not disturbed prior to planting that year's rice crop. So we've identified a number of different management practices that relate to both methane and N2O production. Putting those two together, we would really like to be able to minimize both gases at the same time. Well, that has proven to be very, very challenging because methane and N2O tend to behave in opposite directions. If we try to increase one gas, the other gas is likely going to increase, and vice versa. So it's become challenging to identify one single management practice. But that's what we're working on and striving towards. So there's another twist to rice production that relates back to that water quantity issue that I spoke about earlier. Flood irrigated rice, as you can very well imagine, uses a lot of water. Well, if we can have a production system that uses less water, that might be attractive from a water quantity issue or standpoint. So producers have started to grow rice in an upland scenario like corn, cotton, and soybeans, where rice is then planted on raised beds and furrows next to those raised beds are irrigated periodically. So furrow irrigated rice is gaining popularity in the state without a whole lot of yield loss, but with savings on water and maybe some economic savings from some other labor that's involved in imposing the flood irrigated scenario. So we changed our research approach and started to look at effects of furrow irrigated rice on carbon dioxide, methane, and nitrous oxide emissions. in a full production scale furrow irrigated rice field, we've identified that about 2/3 to 3/4 of the upslope area stays aerobic. Well, then we don't have to worry about methane emissions. But then when we furrow irrigate, we've got this fluctuating soil moisture that tends to increase both carbon dioxide and nitrous oxide production. Now the bottom end of the field, where the water will flow to and accumulate, if it doesn't make its way into a ditch, it might actually back up into the field. And 1/3 to 1/4 of the downslope end of a production scale furrow irrigated rice field tends to behave after a while like flood irrigated rice. It's a little bit of ponding, really wet conditions, that will stimulate more methane production, that will minimize carbon dioxide and nitrous oxide emissions. So the furrow irrigated rice field's even more dynamic, even more spatially variable than a flood irrigated rice system. So that's become equally challenging. We've then moved away, most recently in the last couple of years, to looking at these other upland crops, corn, cotton, soybeans. So we've set rice aside. We've answered and addressed a lot of conservation issues related to rice and air quality. But now we're studying and got lots of ongoing research in eastern Arkansas where we're looking at paired management practices in these other upland crops. So we're looking at the combination of conventional tillage with no cover crop over the winter period compared to a reduced tillage scenario with the use of cover crops. So these two combinations, cover crop and tillage, tends to be an easy practice that producers can use. And so in side-by-side strip trials in producer fields, we're investigating, quantifying carbon dioxide, methane, and N2O production from the traditional management practice, conventional tillage without cover crops, compared to the more conservation-oriented reduced tillage with cover crop scenario. We're also studying the use of another organic soil amendment, biochar. Biochar has gained popularity these days. It's basically a charcoal-like ashy material from the burning of organic material. It might be crop residue, it might be limbs from the timber industry that is just waste material. So it's burned in a process called pyrolysis, where the temperatures are controlled, and it produces this organic amendment, large carbon containing material that has lots of demonstrated benefits, particularly reducing soil bulk density and compaction, increasing porosity, increasing soil water holding capacity and infiltration. But we really have very little idea of the potential impacts of this carbon containing material and its effects on carbon dioxide, methane, and N2O production. We just don't know what to expect until we start doing those studies in the field. And so we've got an ongoing project where we've looked at biochar in a couple of different rates compared to an unamended control. Last year in 2024, we studied furrow irrigated cotton. This year, furrow irrigated corn is being produced. Preliminary results show that there's very little effect on carbon dioxide and methane emissions. However, we've demonstrated that there may be some nitrous oxide reducing capacity from the use of biochar. And so we can't confirm that yet. We got to keep working on our research, getting a few more years of data, but there may be some promise there with the use of biochar to minimize at least N2O production from an air quality standpoint. All of our field research that we've been doing has been using state-of-the-art in-field portable analyzers. We've also have studies where we've got semi-permanent installations where the analyzers stay in the field so we can do more continuous measurements more frequently, literally multiple times a day. And the whole effort is in the direction of trying to quantify carbon dioxide, methane, nitrous oxide emissions from traditional management practices compared to what we might expect from more conservation, sustainable management practices. So let me sum up my presentation this way. The economic, the societal, and environmental importance of agriculture in Arkansas requires researchers and extension personnel to study all of the various conservation issues related to Arkansas agriculture. We've spent a fair number of years studying air quality issues, and we're going to continue to quantify carbon dioxide, methane, nitrous oxide production and release to the atmosphere from traditional and conservation-oriented management practices in an effort to not only maintain high yields and high quality agricultural production from our producers, but at the same time, we want to be protective of our valuable soil, water, and air resources in the state so that future generations of producers can benefit from our high quality resources that we have throughout all of the state of Arkansas. Thank you for your time. I appreciate your attention. We'll answer questions during the Q&A session at the end of this webinar.
All right, welcome back, everyone. I think that certainly gives me peace of mind knowing that even in today's world, these issues with soil and water conservation, we're still doing everything that we can to look into what's happening and trying to find meaningful ways to solve these issues that are in Arkansas row crop production. So I think that is very interesting, Dr. Brye. I do now want to transition us into more hearing from Dr. Della Lunga. Now, Diego, like I said earlier, serves as a postdoc fellow in Dr. Brye's research lab. And since 2018, Diego has been working to quantify trace gases in various row crop production systems using many of those state-of-the-art technologies that Dr. Brye alluded to in his video. And so we're going to hear more about the use of these in-field or in-lab analysis technologies. But first, before we get to that, Diego, I want to know, over time as these technologies have gotten more and more, they've developed more and more over time, have things gotten easier and more simple? Have they gotten more difficult and more complicated? What's kind of been the progression of their use over time?
Thank you, Chandler. I would say both. They got easier, they got more complicated.
Okay.
Instrumentation that we use today, they're built and rely upon mathematical equation that we did not use a decade ago or 2 decades ago. We were not even aware of these possible modeling analysis. Today, the instruments that we can bring in the field are faster, more complicated, but way easier to use. And they can provide us with immediate results in a fashion that even 5 years ago, we were just dreaming about it. And Dr. Brye and I, in the past 5-6 years, we collaborated with several companies to make sure that the protocols that we have and we use in the field are tailored to that specific need.
Well,
reliable results, fast results in the field to address exactly what Dr. Brye was mentioning before and how the degraded soil is going to experience in a fast process the loss of essential nutrients and elements.
okay. Well, it sounds very complicated, but let's go ahead and see what Diego had to say about the use of these in-field measurement technologies and how they've changed over time.
Good morning, and again welcome to the virtual field day. After hearing a lot about the importance of trace gases in agricultural field, now we're going to take a look at various and different analytic techniques to assess these trace gases in agricultural settings. We need to take into account several parameters: agronomic parameters, environmental parameters. Then we have to add the changes in those parameters over time. The good news is that the trace gases coming from the soil surface are a direct product of all these factors at play. So now let's take a look at the first analytical technique that we use to collect and analyze trace gases. And every instrument is indeed divided by two main components, the sampling technique and the analytic technique. The sampling technique regards the collection of the gas samples. The analytical component regards the analysis of the collected gas samples. Okay, what we have here is a component of a system that is called vented non-flow-through closed chamber. This is a collar, this is a cap, we have some vials, a syringe, a thermometer, a portable weather station, and a foam suitcase. The way it works, this collar gets pounded in the ground to a specific depth. You can see here we have 4 drill holes at a specific depth in the collars. Once it's pounded in the ground, the collars will be at the soil surface. This is to allow movement of water inside and outside the collar freely. This is because we want to make sure that the environment inside the collar is exactly the same as the environment outside the collars. When we are ready to collect gas samples, the first step is to plug these drill holes with specific stoppers that will help the system to maintain a sealed environment. Now, in order to create a sealed environment, we have to use a specific cap. The cap has 2 septa on top. The septa allow us to collect the gas sample with a syringe and allow us to insert a thermometer to keep track of the temperature inside the chamber. Remember, idea law of the gas, temperature is extremely important. So when we're ready to sample, we place the cap on top of the collar, and we're going to use a rubber flap to close the chamber and create a sealed environment. Once it's closed, nothing gets in, nothing gets out. We're going to connect specific wires to allow the fan to do its job inside the chamber, mixing the air. We're going to wait a few seconds so that the fan has completely done its job, and we're going to collect several gas samples. Now, the way we collect gas sample is with a 20 milliliter syringe that gets inserted in the septa, and the gas sample is slowly pulled out. Now, the gas sample is transferred to a special vial. This is a 10 milliliter vials. We're going to inject 20 milliliter of gas. We're going to create a pressurized environment in this vial. When we insert the syringe in the vial, you can see that the syringe is kind of moving down by itself. This is because the vial has a vacuum inside. I'm going to talk about this in a second. We're going to inject the entire sample in the vials, remove the syringe, and now our gas sample is collected. In this vial, there's enough gas that allow us to do analysis for methane, carbon dioxide, and nitrous oxide. The vials, once are collected, are stored in a specific suitcase. We call it a foam suitcase, where the vials can be stored upside down to avoid any possible leak. So this technique, this sampling technique, has many advantages and few disadvantages. What are the good part of this technique? Well, this collar can be used in every type of environment. Dry, flooded condition, no problem. That brings 2 main advantages. First, we can have a direct representation of the interaction between biosphere, plants, pedosphere, the soil, and the atmosphere. What are the disadvantages of this system? Well, it's a very laborious and long technique to do in the field. And as we can see in the lab in a second, it takes a long time to complete the analysis. Another disadvantage is that the results from such analysis can be assessed only after the entire procedure is done. So if something went wrong in the field, we will not know until the analyses are completed. This is one of the characteristics that the new system addressed and allow us to take real-time measurement in the field with immediate assessment of the quality assurance, quality control of the data. Now, let's go to the lab to analyze these vials.
And now that we collected the gas sample in the vials in the field, we're ready to do some analysis here in the gas chromatography lab on campus. But before, I want to show you the different components of this system here. We have a series of large gas tanks that we call the carrier gases. We have helium, argon, hydrogen, and air. All these gases are carried through copper tubes into a component here in the system that has a valve and rotors that will direct the airflow and the various gases to the packed column of the system that you can see here in the heart of our GC, and then directed to the different detectors. In this specific GC, that is a 2014 Shimadzu, a company from Japan, we have 3 different detectors, as we can see on top of the instrument. We have what we call a flame ionization detector, we have an electron capture detector, and we have a thermoconductivity detector. Now, our specific GC is also equipped with what we call an auto sampler. It's a robotic arm that allows to perform analysis on a continuous basis. So our vials that have been collected in the field, now ready for analysis, are going to be placed on trays. We can have multiple trays in the auto sampler, and the robotic arm will collect 1 millimeter of gas sample from each vial, injected in the port here of the instrument, and then it will be directed to the various detectors. The analysis of vials with this system are particularly long. Each vial can take up to 18 minutes to complete analysis through the 3 detectors. So one of the disadvantages of this system is that the operating procedure can be quite long, and if we have several fields and several studies, the instrument might be working for days before you can take a look at the data collected in the field. However, one of the great advantages of this instrument is that the detection limit is extremely low, so allow us to be extremely accurate in our analysis. So while precision was something we wanted to have in the field as a sampling collection, when we are back in the lab, accuracy becomes more important. And with this, we conclude the part with the gas chromatography technique, and now we can take a look at portable in-field instruments that allow us to do the same job remaining in the field.
And we're back in the field to take a look at a complete different system to analyze trace gases from the soil. What we have here is a complicated system that uses a method that it's called enhanced cavity absorption with optical feedback. What it means is that we have analyzers that inside they have a chamber with a laser, a mid-infrared light that is shot between highly reflective mirrors. When a molecule of trace gas goes through this infrared, a portion of that light is absorbed, and is absorbed at a specific wavelength and with a specific pattern, specific to a molecule of gas. So when the infrared light comes out of this chamber, it will hit a detector that records how much light, how much infrared was absorbed, and with which pattern. and we record those data and transform these data into a concentration of a specific trace gas. Now what we have here is a system from Lycor, is a company in Nebraska. We have two analyzers, a series of tubes, and what we call a survey chamber. So the principles are exactly the same. We have a chamber that is going to create a sealed environment once it closes. Tubes that will direct the airflow with the gases into the analyzers. The analyzers will analyze the samples and report the gas concentration in real time. Well, let's take a look at the analyzer inside. Here we have different components. We have a slot for the batteries that will power the analyzers for about 8 hours. A small computer that records the data, the concentration over time, and then can communicate with the other components of the system through Ethernet. Here we have our chamber, our cavity, where the magic happens with our infrared and the reflective mirrors. And then we have a series of tubes that go through a desiccant gel. This part removes almost entirely the water vapor in the system. So we remove the water vapor, but we still keep track of the water vapor in the system so that we know that nothing gets damaged. Now, the collars that we use for this system obviously is going to be smaller. It's going to be the size of the chamber. There is a gasket here that will grasp on the collar in the soil. The rules are the same. The collar is pounded into the ground to a specific depth in order to create a sealed environment. Now, this entire system can be controlled remotely with an iPhone, a tablet, a laptop, computer. So how long do I need to keep the chamber closed? Where this specific system is able to report the concentration of trace gases every second. So you can imagine that because we're still using the best fit of a linear regression among points over time, we don't need 1 hour of observation length. So recommendation from the company and field trials report that an observation length of 5 minutes, it's more than enough to capture the realistic trend, the reliable trend of trace gases from the soil. Once the observation is completed, data are extracted from the chambers and from the analyzers with the software that we call Soil Flux Pro that allow us to perform all the calculation that previously we were used to do by hand. So there was a lot of room for error. Now everything is done with a specific software. So this system has a lot of advantage and few disadvantage, as all the system we're going to see today. What are the advantage of this system? Well, this system can provide real-time data, so we know what happens in the soil immediately. So we know that if something is not working, if data are not representative of the condition that are in the soil, we know that something went wrong. We can take care and change the setting right away. We have multiple gases that can be analyzed at the same time. There are other type of analyzers that can do even more gases. One example is ammonia. And is portable in the field. What are the disadvantages for this system? Well, this system cannot be used in all type of environments. If we are in flooded condition, this system will not work. The water vapor and the molecule of water that can be captured in the system are too dangerous for the system to work, to be used in that environment. Another disadvantage with this system is that the chamber is very small, and you can see doesn't allow for much room inside. So collars that we use for this specific equipment will not have plants inside. These conclude the cavity enhanced absorption spectroscopy. Now we're going to take a look at something similar but yet different. And what we have here is a different type of instrumentation that still uses infrared spectroscopy. We have an analyzer, GT5000 Terra, and a closed chamber again. The analyzer comes from Gasmet, a company in Finland that developed the smallest trace gas analyzer in the world. The chamber that we use for this specific system, you guessed it, is the Lycor chamber as we've seen before in the previous systems. The concept is the same. There is an infrared laser shot in a chamber inside the analyzers between highly reflective mirrors. Now in this specific case, the mirrors are coated with gold, and one of the mirrors is moving. It's fancy. This allows to enhance and enlarge what we call the light path of the laser inside the analyzer, that in this specific case reaches 5 meters. So the light is bounced between these moving mirrors, creating a 5-meter path. The molecule of gas going through this 5-meter infrared path will absorb some of the wavelength, with specific frequencies and patterns. Now the analyzers uses what we call a Fourier transformation. This analyzer indeed is called FTIR, Fourier Transform Infrared Spectroscopy. The Fourier transformation refers to the angle of the mirror and the path of the light to convert an interferogram, so an absorbance spectrum detected by the interferometer inside, to absorbance spectrum, where concentration can be derived. Complicated mathematical equation that likely the software does it for us automatically. Now by this point you might wonder, what is the small thing next to the chamber that we've seen before? This is a sensor. The chamber from Lycor allows to have several sensors installed. This specific one is called Hydra probe. Can measure volumetric water content, soil temperature and electroconductivity once inserted in the soil surface to a depth of 6 centimeters. The important thing is that this metal plaque at the bottom of the Hydra probe is in contact with the soil surface. So this system, as well as the Lycor system, allows to keep track not only of trace gases but environmental parameters that can be associated to production and release of greenhouse gases. The system has different quality assurance and quality control parameters. Specific, we have what we call the background control. Before we start the analysis, what we want to analyze is basically air that is pure, that has no trace gases inside, so that we can look at the interferogram and see that there is no contamination from trace gases. The way we perform this background is again flushing N2 into the system, so we do not allow any air from the field to come inside the analyzers. What we want to see is a complete clean interferogram where we do not have any peak of trace gases. Once we have OK from the interferogram, then we can proceed to the analysis. Now the entire system can be controlled with a tablet remotely. They use a specific software called CalcMet that can show in real time the concentration of the trace gases collected every 20 minutes, along with different parameters of the instruments, temperature and water vapor. As we mentioned before, the system is very sensitive to water vapor, but also is very sensitive to temperature. So here in Arkansas, during crop production, during growing season, we can have temperature in the fields that are 110 degrees. And if you ever spent a moment in a cornfield, you know that that number goes up as soon as you step between the canopy. So how do we manage that issue? Well, we built this wonderful structure that gets placed on top of the instrument before the analysis. This highly reflective tape reduces the temperature that is, and the solar radiation that is reaching the instrument, limiting the increase of temperature that we observe in the field. Now, my nephew did not do this, I did. I'm not very good at art and craft, still works. What are the advantage and disadvantage of this system? Well, again, we have real-time analysis, so we can see the quality of the data in real time when we are in the field. This analyzer can measure up to 50 different gases, so we have a great versatility in the field. We can perform a large series of assessments in terms of trace gases. What are the disadvantages? Well, the observation length is longer, so we have to spend more time in the field, and the system is very sensitive to water vapor and temperature. An additional disadvantage is that we need to carry highly pressure gas tank in the field that in some cases can be challenging. And now we're going to take a look at the last type of equipment to keep track of trace gases in the soil. We made it to the end. We're going to take a quick look at yet a slightly different equipment we use in the field. Here we have what we call a smart chamber, still a Lycor chamber. Now I need you to use some of your imagination because I couldn't bring the entire system here. But try to think about solar panels, rechargeable batteries, an entire system connected to analyzers from Lycor, a central computer, and few of these smart chambers. Now this system is completely automatic. This system basically operates on a continuous basis every day, multiple times a day. The way this system works is that the central computer tells the various chambers in the field to close at specific time. We have this robotic arm that will close on the chamber, on the collar here, creating a sealed environment. This chamber will operate with the same settings and parameters that we described for the Lycor system we've seen before. And all the data will be stored in this central computer. The system can also be equipped with an intracellular modem that allows us to connect remotely. So we can change parameters, control the data, and extrapolate the data from this system. All the rules we described before still apply, but I wanted just to show you how the advancement in technology is allowing us to do more work with less time spent in the field. And with this, we conclude the section about all the instruments for trace gas analysis that we have here at the University of Arkansas in Dr. Brye research team. So what we learned and observed today is that while there are still many questions that need to be addressed about biogeochemical cycles in the soil, the technology that we have today, the technology already deployed in our fields, will allow us to have reliable results, an essential element to provide management practices for our farmers that will keep those biogeochemical cycles in equilibrium to maximize crop production for long-term sustainability. And with this, say thank you for your attention and back to the studio.
All right, perfect. I work with these technologies as well. I like knowing, the background. They're getting better with time. We're getting better and better with the data, getting better at working in the fields. So thank you, Diego, for that insight.
Pleasure.
And so I think with that, we can go ahead and transition from our presentations into the actual Q&A of today's presentation. So I think we'll start with questions that have been asked from the chat. looks like our first question, and either of you guys feel free to answer these, but the first question was, with furrow rice, did you notice having an increase to herbicide and insecticide applications?
I'll defer that one to Diego to address.
Dr. Brye and I spent 3-4 growing seasons studying full irrigated rice, and although the economic analysis of the input cost was not specifically addressed, we did realize that the weed pressure in a full irrigated rice was going to represent a potential issue. An issue that, if you think a few years ago, the only solution was to apply several products as herbicides during the growing season. As technology is moving forward, we see now that pre-emergent application of herbicide in a full irrigated rice field, specifically in the up mid portion of the field, might do the trick if it's done at the right time.
Okay, yeah.
Yeah, one of the major advantages of having the flood is actually weed control. There aren't many other plants. There are a few of them, and they are issues in flood irrigated rice production, weeds particularly, that can tolerate those conditions like rice can itself. But when you release the flood, now you basically are opening the door for lots of other potential weeds and pests.
Yeah, that makes sense. Looks like we have another question. Going from soybeans to rice to soybeans in rotation, do you notice a loss of soil in preparation of the field for each of these different crops in rotation?
Well, when you've got this type of a rotation, the rice-soybean rotation, remember any sort of irrigation management works well when you've got relatively flat terrain. So we're not talking about having real steep slopes where gravity is assisting with potential erosion and water runoff. It also goes back to the choice of tillage practice that is used in that rotation. You know, if there's conventional tillage that is used and the spring of the year prior to planting a particular crop, the soil is disturbed, yeah, you can expect to lose some soil off the field, perhaps into the ditch, during rainfall runoff events or irrigation events with excess water. But if you've got a reduced tillage type of scenario, maybe even a no-till, that's going to do a really good job at keeping the soil in place on the field and basically stop it from moving off-site.
Okay, yeah. We have a bit of a longer question here, so I'll go slowly with it. But do you think using biochar to absorb nutrients from manure, wastewater, leachate, something like that, could be an effective solution for nutrient cycling, reducing trace gas, greenhouse gas emissions associated with manure or synthetic fertilizers, especially if you then used this nutrient-rich biochar as a fertilizer?
I'll go ahead and start, and I know Diego can follow up with some thoughts. This is a very interesting concept. Waste materials, certainly those that are liquids, there's the potential to filter nutrients out. Biochar has a filtration capacity. This is somewhat analogous to using charcoal in a fish tank to help filter out impurities. There is some application that's already been used in household wastewater remediation, where household wastewater is moved through basically a box containing organic material. Might be charcoal, might be something else. And then applying that sort of a concept, a technological innovation to an agricultural liquid waste material makes some sense. I'm not personally aware of anybody that's actively doing that, at least at the University of Arkansas. But that would be something to me that would be worth an interesting look into. And then you make sure that those materials are then land applied, and there might be some long-term benefit to a production system. Diego, what are your thoughts?
It's definitely an interesting concept, and the idea that we can have a double effect, that we're basically extracting nutrient from the manure and then reapplying in the field with biochar poses some serious challenges. the biochar has a great absorptive capacity, so can retain this nutrient fairly quick. The release of nutrient from the biochar as applied, we might think might be slower. So when we budget this nutrient as input in the field from an enriched biochar material, we have to keep in mind how the cycle will work over time, what will be the timeline. And to have those questions, we definitely need to study it. So maybe next proposal.
Yeah, certainly. Yeah. Someone asked, so if I understand correctly, there is no current data on tillage no cover crops versus furrow cover crops on decreasing gases. So I guess the comparison of tillage with no cover crops versus, I guess, a furrow raised bed system with cover crops on its effect of decreasing trace gas emissions.
Diego, I'm going to defer that to you.
What kind of?
So right now we have 8 study sites where tillage and cover crop and some combination of these factors are being evaluated in comparison to conventional practices. Maybe just tillage, maybe tillage with no cover crop, different type of depth for the tillage. The answer will be based on 2-year study, so next season we will have reliable answers, and might not be what we want, but they will be based on ground truth data. We have to keep in mind that when we study conservation practices, the answer that we seek, it is not hidden in a short cycle of 1, 2, or 3 growing season. The real nature of that management practices is revealed after a long period of time. What we can do in the short period of time is to make sure and to assess that we are in the right direction, although those results might not be on the scale and magnitude that we're hoping and looking for.
So at the moment, I don't think there's a whole lot regarding every single management practice combination, especially when you start tacking on tillage plus or minus a cover crop, and there are other management practices you could throw into the mix there. This is an evolving science, and so greenhouse field studies, proof of concept needs to happen for a lot of these different combinations. We're doing what we can with the resources that we have, and as Diego alluded to, stay tuned. There'll be more to come from my students, my research group, from a publication standpoint in the next coming years.
Excellent. It looks like we have another question specifically for Diego. This person asked, Dr. Diego, which greenhouse gas analysis method do you prefer? Gas chromatography, the use of LICOR, or the use of the GasMat system? And what factors influence this preference in the field, in the lab? I mean, they take you lots of different places.
Definitely a question where diplomacy will be used. I definitely recognize that there are characteristics of the system that can be better tailored to specific studies. Now, we work in a little bit of a backward process. We have this equipment available in our lab, and we have been trained to use these systems in different conditions. But we basically develop studies where we can use this system for that specific purpose. I'll give you an example. We have studies that we conducted on greenhouse settings. Mm-hmm. And we really like to use the gas chromatography in our closed chamber made with PVC for that specific environment. And then when we go into the field, we know that we prefer to have a portable instrument because we will have real-time data. So it really depends about the conditions of our experiment. But when I started my graduate program with Dr. Brye, I do remember working in rice fields where we were carrying PVC extenders for our closed chamber, 20-30 pounds each, for a mile into the field, several trips
Wow.
per sampling time. Man, that was a workout.
A lot of labor.
And I'm glad now we can all put everything on our backpack and just walk into the field.
And I think you said it in your video, but also you've talked to me about how, gas chromatography, you're able to include plants. So yeah, it certainly seems to depend a lot on, what do we want to measure? What kind of crop do we want to measure it in? So absolutely, I think each one has its pros, its cons.
No doubt.
Absolutely. Let's see. Our second to last question, it looks like, is specifically for you, Dr. Brye. Say, personally, what inspired your decision to pursue a career in agronomy and soil science? And what aspects of this field continue to motivate your work forward today?
Well, I've got a very specific response for that question because there is some history there. I didn't grow up on a farm myself. I'm native from southern Wisconsin. Both of my parents grew up on farms. I've spent a lot of time on farms, particularly on my father's side. He grew up in west central Wisconsin. And as I mentioned in my presentation, soil erosion was a massive problem decades ago, all the way throughout the country. And the tiny little town, 800 or so people as a population, was an epicenter of one of the biggest, most successful watershed scale field projects, demonstration projects to combat soil erosion and water quality issues. And that was centered in the small town that my father grew up on the farm with my grandparents and his brother and sister. So there's a state of Wisconsin historical marker on my extended family's farm. It's still there today, commemorating the Coon Creek Watershed Project. And that is literally the inspiration that I've had when I was an undergraduate student. I changed my major from wildlife biology. I wanted to be a game warden, changed it to soil science, and haven't looked back since. Nearly on a daily basis, I learn new things. I really had hoped I'd never saturate my knowledge, and that has absolutely come to fruition. Working with all the various students I've had over the years, and currently there's new and exciting things that I really think that that's the inspiration for my personal career and is motivating me to continue on, just trying to do what we can for conservation of all sorts of resources, and I'll look forward to sustainability for future generations.
Okay, yeah. So the roots of your soil science background are deep.
Roots have run deep.
They
Absolutely.
We got another question. I said the last one was second to last, but maybe this will be the second to last. Someone asked, and Diego, both of you would probably answer this. Funding is a big thing in academic science. So could you talk a little bit about, the different financial aspects and inputs for these different trace gas analyzer systems? That no doubt they're expensive, but, how do they differ in our ability to fund their operation?
The equipment, the analytical equipment we use in the lab, Diego showed the gas chromatograph, the various field portable analyzers we use in the field, the long-term system, they are expensive. And that is, frankly, a big burden. We have to be able to find the resources. Luckily, we've been successful recently to be able to build our analytical capital in our lab. We baby these instruments. We need to make sure that not only are they functioning now for the current projects, we don't break down, we don't end up having to miss field measurements. So we're very proud that we're able to do as frequent of in-field measurements as we can. But gaining resources to do the type of research, air quality, conservation work is definitely a challenge. We have to seek out specific opportunities and roll the dice. We try to, with our words on paper, sell what we have, and we've done a pretty good job so far, and we want to keep doing our high quality work. We want to take what we've started all the way to the end. We want to publish our work and contribute to high quality science, because that builds our reputation and allows a funding agency, a funding group, to be very confident in where they might be putting their resources.
Yeah.
And if I may add,
Yep.
another aspect to consider is that no doubt the investment is substantial to do this type of work. But as Dr. Brye was mentioning, if you work on these machines on a constant basis, the maintenance cost is reduced almost to nothing on a growing season basis. So for a growing season, the cost of a maintenance for this, specifically for the portable analyzer, if you're very careful and you maintain the status of the equipment properly functioning, the cost of maintenance is substantially reduced. And these companies that provided the instrumentation and we purchased from them, they work constantly with research teams to make sure that everything is working correctly and every single thing can be addressed in a matter of like hours.
Oh, okay. Well, that's excellent to know. I think with that, that's all of the time that we have for questions today. I want to thank the both of you for being here today and offering up your expertise. I know you have very busy
Thank you.
schedules. So,
Absolutely, our pleasure.
I appreciate you guys being here. I want to thank the USDA and RCS for also sponsoring this project and collaborating with us. I also want to thank my other colleagues here at the University of Arkansas Division of Agriculture for helping us organize this series and putting things together like this lovely studio. And I want to thank you, the audience, for taking interest in these topics and caring about our environment, our soil and water conservation series. I mean, agriculture affects all of us every day. And so these topics and these problems, they have to be solved. Someone has to do the work. So I want to thank you all for, tuning in and hearing what we had to say. So with that, be on the lookout for future communication from our team. We will have more virtual field trips in the future. We have another on July 7th looking at blackberry production here in Arkansas. So be on the lookout for communication about that. And I think that's all that we have on our end. Again, thank you for tuning in, and we'll see you next time.
Thank you.
Thank you for joining us for the Soil and Water Conservation Virtual Field Trip. This broadcast is funded by NRCS and produced by the University of Arkansas System Division of Agriculture Cooperative Extension Service.